Testing, testing, testing. Oh, McKenna, you guys are doubling up. Where are you guys? You in the lobby or something, or?
We're in the library, in the study room.
Oh, I heard the internet in the library wasn't very good.
Worked for us so far.
Oh,
Already
okay.
got one class.
Okay. Hey, works for me. Hannah, that wall needs something behind it. It needs a picture or a tapestry or a, looks very bare.
You're right, it is.
See, look at Jessica. Look at that beautiful wall. All that stuff on there. Kind of looks like my office. Sterile. That's why I don't do this in my office. Okay, try this. All right, that works. Yeah. Let's try this. And that works. All right. All right. Up there. Hey, Natalie, does the weather affect your basketball schedule? Natalie, can you hear me?
Well, people are just getting up.
Oh, okay. Natalie, does the weather affect your basketball schedule?
Yeah.
It does? Okay, just keep me in the loop as to when you miss class. Well, Mickey says it's 1:30. So, and I heard old big BB out there chime, so I guess it's 1:30. So we're gonna go ahead and get started. Again, I know you guys hate Zoom. You've told me that before, but who am I to essentially tell the administration to go pound sand? So we're doing this via Zoom. We will be in class as of tomorrow. I've been told we'll definitely be in class on Friday. So you guys don't get out of the quiz that easily. So there we are. Okay, well, let's have a word of prayer, then let's get started. Father, thank you for this snowy day. We pray as we look out and see the field of white, we understand that you have made us as white as snow. We thank you, Lord, that it is not with perishable things such as silver and gold that we were redeemed from the pointless way of life handed down to us, but with the precious blood of Christ, who is a lamb without defect or blemish. Thank you, Lord, for the message of the white snow. And now, Lord, on the strength of that message, help us now learn about chromosomes, DNA, and the like. In Jesus' name, amen. All right. We looked last time at chromatin, and we put together some models. And we saw that the nucleosome consists of these 4 different histone proteins, H2A, H2B, H3, and H4, that are put together into an octamer. There's 2 copies of each of those proteins. They form an octamer. The DNA winds about each of the octamers around about 2 times, roughly. These are then wound into a tight spiral like this. You have this structure called a solenoid. The alternative structure is a zigzag model, but virtually all the crystal structure data says that this model is right, the solenoid model. Solenoids are very dense, not even water can get in between them, it seems. They're so dense. And they form a thread, and you can see they are 30 nanometers in diameter, well, in the width across these is 30 nanometers. That's why they tend to be called a 30 nanometer thread. All right, a word about structure in the nuclei, in a nucleus. So let's go to our makeshift blackboard here. All right, so let's pretend this is a cell. And we have a nucleus here. Oh, that yellow doesn't come out very well, does it? That's hard to see. Let's use a color that's a little easier to see. Let's maybe use that. Okay, that's probably easier to see, yes? So that's your nucleus. And this over here, of course, is the cell membrane. And this, of course, would be the cytoplasm here. And we're going to take a look inside this nucleus. We're going to kind of blow this up like that. So if we blow up the nucleus, so we're taking the nucleus, we're increasing magnification of the nucleus. On the outside of the nucleus, of the nuclear membrane, we have this structure that is a series of proteins. There's 3 proteins. They come together, they form this matrix just underneath and my artistry, as you can see, leaves much to be desired, but I hope this gets you at least an idea of what's going on here. These proteins underneath the nucleus, they are called nuclear lamins. So they lie underneath the nuclear membrane and they provide structure to it. Nuclear lamins. There's 3 nuclear lamins and they're called nuclear lamins A, B, and C. So these lamins essentially are going to, they sit right underneath the membrane. If these get disrupted structurally somehow, the nuclear membrane falls apart. In fact, when you get into prophase of mitosis and the nuclear membrane breaks down, that's how it breaks down. These nuclear lamins are disrupted. So also within the nucleus, you have a filigree of proteins that form little fibers. Like this. These fibers within the nucleus, they are collectively known as the nuclear matrix. The nuclear matrix. So just think of the movie, The Matrix. Not the redos. They were terrible movies. The original one, which is really the only good movie out of the lot. These nuclear matrices consist of 8 different proteins that are given the incredibly creative name of matrins. There's 8 of them, 8 different types of them. And they come together and they form these fibers. Now your chromosomes, as it turns out, are going to be organized around these fibers. and so on and so forth. So the chromosomes don't just float within the nucleus. They're actually, there's a house, if you will, for them to live in. And that house consists of these nuclear matrices, and they're held in place essentially by the nuclear membrane, and the nuclear membrane is supported by these nuclear lamins. And we could draw all 23 chromosomes in there if we had time, but we won't. So the 30 nanometer fiber is going to bind to the elements of the nuclear matrix. Now, the nuclear matrix in general consists of the nuclear lamina itself that we talked about, that fibrous, the nuclear lamin A, nuclear lamin B, nuclear lamin C, laminin A, B, and C. They form that filigree underneath the nuclear membrane that supports it. And then inside the nucleus, you have the nuclear proteins. And it's very clear that parts of the genome bind to the nuclear matrix, in some cases pretty tightly. Now, the DNA binds to the nuclear matrix in the form of loops. In the form of loops. To illustrate that, I'm going to once again draw you some more of my famously bad drawings. But I hope they help. I hope they give you an idea essentially of how this works. So essentially, you have, let's say, a internal nuclear matrix complex, and it forms essentially this fiber. If we take the fiber and we do a cross section of it, it looks like that. And then maybe we can swing it around this way, and it goes that way, and something like that. So that would be one of those fibers made of those matrins and some other proteins. Now your DNA, oh, what color should we use? Let's use this one. Your DNA, your 30 nanometer fibers are going to bind to this, and they're going to form a loop like that. Then there's going to be some more of this, and they're going to form another loop kind of like that. And there's going to be more, and they're going to form another loop kind of like that. And there's going to be some more, form another loop kind of like that, be some more, and then another loop, and it's going to go like that. And then you'll have essentially, this kind of looks like a pinwheel, doesn't it? And essentially forms a pinwheel all along that fiber. So essentially you get kind of a fiber with these pinwheels on it like that. Now, this structure, as it turns out, is 700 nanometers in diameter. Now, you might be sitting there writing this down saying, why is he telling us this? I'm not going to remember this. This is about as important as a dead frog on a rotting stump in a swamp. Well, it turns out a metaphase chromosome is 1400 nanometers in diameter. Since when you have chromosomes at metaphase, you have two chromosomes next door to each other, this structure, because its dimensions fit so beautifully with what you see with a metaphase chromosome, that is the width of a metaphase chromosome. That tells you that this structure that's been made in the laboratory and has been seen using high resolution electron microscopy, this is real. It's not an artifact of preparation. It's not an artifact of the test tube. It's actually real. And these loops, you can actually see these loops. In fact, I'll show you a slide here in just a bit where you can see those loops pretty easily. So the chromosomal DNA is arranged in those loops around the internal nuclear matrix fibers. That helps organize the chromosomes in the nucleus. And as it turns out, each chromosome is assigned a place in the nucleus. It's like each chromosome has an address in the nucleus, and that little area in the nucleus where it's assigned is called its chromosome territory. So here's a picture that looks a lot better, of course, than anything I can draw. And this is meant to illustrate the nuclear lamins. And the nuclear lamins, let's see if we can get. So the nuclear lamins, as you can see, are here, just inside the membrane. And then the fibers, as you can see, are all in here, like this. And then these yellow globs, those are meant to represent these proteins that are known to bind to the nuclear matrix. Interestingly, there are several genetic diseases that are known to result from loss of function mutations in elements of the nuclear matrix. Have any of you ever heard of an aging disease, premature aging disease called progeria? That's P-R-O-G-E-R-I-A, progeria. So babies, when they're born, by the time they're about 4 years old, they have severe wrinkles in their skin and their arteries are clogged with atherosclerosis, just like in an older person. These people usually die in their adolescence years from old age. It's a premature aging disease, and it maps to mutations in the nuclear lamins. So if I convince you that the nuclear matrix is important. Oh, that's okay. Your stunned silence tells me everything. All right. Again, if you guys have questions, please chime right in. If you use the chat function, I may not see it. So just unmute yourself and just ask me a question. If you're not talking, though, please mute yourself. Otherwise, we'll get feedback through your speaker picking up me and then feeding back into my speaker. Please don't ask me to explain how that works, but it does. Now, these loops, as you can see here, there are portions of the go back to our, there we go. There are portions of the DNA that actually bind very tightly to the matrix, and these are called scaffold attachment regions, or SARs. So if you take the genome and you chop it up into little pieces and you give it to isolated nuclear matrix proteins, only particular portions of the genome will bind to the nuclear matrix. These loop domains, they're anywhere from 25,000 to 200,000 in size, so they vary in size pretty substantially. And the bits that are bound to the nuclear matrix, as you can see here, these are these matrix attachment regions or scaffold attachment regions. They attach to the nuclear matrix. These sequences have been isolated. They're very AT rich, and they're not terribly well conserved. Why they attach to those proteins is not completely clear, but they do. So this, I just think this is amazing, but this was first found in the fruit fly. So the fruit fly only has 4 chromosomes, and the salivary glands of the larva of the fruit fly have these very, very, very large polytene chromosomes that are very easy to see. They're so big, you can see them very easily with a light microscope. If you grab the mouth parts of the larvae and you pull, the salivary glands come with it. You can then soak it in a mixture of acetic acid and lactic acid, and then you put it under a siliconized cover slip and you press the bewilders out of it, and the chromosomes will spread really beautifully under preps like that. And if you do those preps all day, you get a very sore thumb. Anyway, if you do those preps, but you leave the nuclei of the salivary glands intact, and then you use specific probes for specific chromosomes, the same part of the nucleus for, say, chromosome 2, for the right arm of chromosome 2, the same region of the nucleus lights up in every nucleus you test. And then if you use a specific probe for the left arm of chromosome 3, the same region of the nucleus lights up in every single cell that you test. And same with the X chromosome and the chromosome 4, which is very small, and so on and so forth. And then when this was extended to the mouse and then to human cells, it turned out to hold. It turns out that the chromosomes are not haphazardly arranged in the nucleus. It turns out they're arranged in the nucleus in a very highly ordered and reproducible fashion. And this picture here illustrates essentially probes from many different chromosomes that are labeled with fluorescent dyes that produce different colors when they fluoresce under a argon laser. And you can see if this is the way this looks in virtually every cell you examine. Chromosome 4 is always right here. Chromosome 1 is always over here across from chromosome 4. And so on and so forth, and blah, blah, blah. You get the impression here. So that's the case in an interphase chromosome. Here are your metaphase chromosomes, again, also lit up with this technique. This technique is called FISH, fluorescence in situ hybridization. Fluorescence in situ hybridization. You probably want me to write that, don't you? Okay, I'll write that. Let me go to our, no. Yes, discard. I don't want to keep those. Go to our makeshift blackboard. I'll write that down. So that's fluorescent in situ hybridization, otherwise known as FISH. So you take let's say this is a gene, and you know this gene maps to a certain chromosome. You then simply, you put this in a test tube and you use a kit that will replicate the DNA, except you use nucleotides that are labeled with a fluorescent dye. And then depending on the chromosome, you do this with genes that label multiple different chromosomes. And every time you do this, you use different nucleotides that you can buy that have different dyes that will make different colors when they fluoresce. You put them all then in a test tube, you denature them, and then you hybridize them to your chromosomes using protocols that are widely available, that are very well worked out. And you get those kinds of pictures essentially that you see in the slide that I just showed you. FISH. Very labor intensive, but very powerful technique. And this, as you can see here, these chromosomes are all different colors because they've been labeled with different probes. And those various probes essentially will light up, hybridize and light up each chromosome when you shine it with the right colored light. And this person here, Irena Solove is one of the pioneers of this technique. And her postdoc, Felix Habermann. Felix, I think, is now at EMBL, which is in Heidelberg. And he's doing a lot of interesting cytogenetics as well. Now, some DNA is very heavily transcribed. The genes in that region of the genome are being transcribed, and they're being transcribed often. So that portion of the genome is not wound into tight chromatin. It's wound into relatively loose chromatin so that the transcriptional machinery has access to it. That type of DNA that is heavily transcribed, we call that euchromatin. Euchromatin. It's transcriptionally active. You have the 30 nanometer fiber. It forms these loop domains, but they're fairly loose. They're not tightly compacted. And this is less condensed regions of the chromosome. There are other portions of the genome where the DNA is not transcriptionally active. It's transcriptionally inactive. That DNA is wound into very tight, highly compacted chromatin. And the 30 nanometer fibers are wound into loops that are smashed together essentially into very tight structures. And this type of structure is called heterochromatin. Heterochromatin. So heterochromatin, not transcriptionally active, very tight chromatin structure. Euchromatin, transcriptionally active, not tight chromatin structure. And there are stains you can use for chromosomes that will stain euchromatin more darkly than heterochromatin, and vice versa. That's how you can discover what part of the chromosome is what. Now, if you use such stains, there are two things that kind of fall out. Well, three things that fall out pretty routinely. Number one, the telomeres and the centromere are consistently heterochromatic in every chromosome, all 23 of them. So the centromere and the telomeres are heterochromatic. So they're wound into tight chromatin. They're transcriptionally pretty silent. Between the centromere and the telomeres, you have heterochromatin that is scattered, but that heterochromatin pattern differs from one cell type to another. That heterochromatin pattern differs from one cell type to another. Therefore, you have some regions of the genome that are heterochromatin in every cell. We call such regions constitutive heterochromatin. Constitutive heterochromatin. You guys know what constitutive means? Does anybody not know what constitutive means? Okay, McKenna's brave. She raised her hand and said, can you please define it for me? Constitutive means that's the way it always is. So let's say Sydney is just, she's constantly hungry. She always has to have snacks with her. She's constantly munching on something. Her metabolism is just very, very fast. And so she carries a box of crackers with her wherever she goes. She's always hungry. That means Sydney would be constitutively hungry. Constitutively hungry, constitutive, it's that way all the time. These other portions of the heterochromatin that vary from cell type to cell type, that's called facultative heterochromatin. Remember last semester, we talked about facultative anaerobes? They can grow either in the air or they can grow in the absence of air. So they can do one or the other. Facultative heterochromatin. Sometimes in one cell type it is heterochromatic, in other cell types it's not. So for example, if you look at a liver cells make lots of proteins that are secreted into the blood. Many of the genes that secrete those proteins are very transcriptionally active in liver cells Some of those genes are clustered together even in parts of the genome, and those regions are euchromatic in liver cells. However, if you go to a neuron, those regions of the genome are wound in heterochromatin. So they're heterochromatic in neurons, but euchromatic in liver cells. So that's an example of facultative heterochromatin. It's heterochromatic in one cell type, not another. Okay, good. Let you finish writing. So this is what's meant by these two types of heterochromatin. And I really like this picture here on this slide because it shows how the centromere and the telomere are in fact heterochromatic, but these other portions that aren't wound as tightly, they're euchromatin. And then some of these bits will be heterochromatic, but it'll differ from cell type to cell type. And those will be facultative heterochromatin. They can interconvert between euchromatin and heterochromatin. In fact, one good example of this, if you look at a very, very young thymocyte, so a pre-T cell that is making its way from the bone marrow to the thymus, there are regions of that cell that are wide open, transcriptionally active. Once that cell becomes a mature T cell and becomes either a helper or a T cytotoxic cell, or maybe a suppressor T cell, or some other type of T cell. Now, many of those regions that were transcriptionally wide open are now cinched down and are heterochromatic. So they went from euchromatic to heterochromatic as the cell now became more mature.
So with that picture right there that you just had on that slide,
Yeah.
would that have been a facultative because it goes back and forth between?
Yeah, but this part here at the centromere and at the telomere, that's constitutive because it's heterochromatic in every cell you look at.
Okay.
Just a tidbit, I guess, of potentially worthless information, but some of you are interested in peds and neonatal medicine and OB and what have you. So embryonic cells, when you look at embryonic cells have very little heterochromatin. Their DNA is wide open. As those cells start to differentiate and start to become certain types of cell types, now regions of their genome start to get shut down. And the regions that get shut down essentially cut off the types of cell fates that that cell can achieve. And as the cell becomes more and more mature, more and more portions of the genome get shut down. So that's one of the ways that cells that are very naive essentially become very differentiated, is portions of their genome now become shut down. So as cells enter mitosis, M phase, the DNA now becomes dramatically more compacted. So by the end of prophase, chromosomes you could not see during interphase now condense. By condense, we mean they're wound into tight heterochromatin, and we can see them quite nicely. And so you get two chromatids next door to each other to form a single chromosome that have a diameter, a width of 1400 nanometers. And they're quite a bit shorter than they were in interphase. And metaphase chromosomes don't undergo a whole lot of transcription. That's why when the cell enters M phase, it has to have made everything necessary for mitosis. So the levels of compaction that I already shown here, there's your double-stranded DNA double helix. Those are then wound about histones to form nucleosomes. The nucleosomes are compacted by H1 into a 30 nanometer solenoid. They form a 30 nanometer fiber. Those fibers then form these radical loop domains. Those radical loop domains have a length average of about 300 nanometers. Those will then arrange on the nuclear matrix thread. That nuclear matrix thread then forms a structure with a width of 700 nanometers. You put those side by side on a metaphase chromosome, you get a structure that's 1400 nanometers. And I need to emphasize, this looks very 2D. This is actually a 3D structure. So think of this as a rod with loops kind of coming all around it like that. This is not a 2D structure, it's a 3D structure. During metaphase, the chromosomes are linked to scaffolding, and the scaffolding is formed from the nuclear matrix, the internal nuclear matrix at least. In some cases, some of the histones are specialized for certain parts of the chromosome, in particular the centromere. Those modified histone octamers are necessary for the assembly of the kinetochore. Here's a beautiful picture of a metaphase chromosome. Now, if you treat this with high salt to remove the histone proteins, I don't know if you can see that terribly well, but do you see the loops of DNA? They're tight, packaged tightly together. Some of them around here, do you see the loops? I wonder if I can get that to look bigger. Oh, that better? Can you see the loops there? Yeah. So those are the loops that are associated with the nuclear scaffold. When I was in graduate school, the nuclear scaffold was a subject of some debate. There were lots of people who thought that this was an artifact of preparation. And you had dueling cell biologists essentially going back and forth in journals, one arguing that the nuclear scaffold didn't exist, the other arguing that it did exist. The did exist people won the argument. There's very, very good evidence now that the nuclear scaffold does in fact exist and is exceedingly important in the life of the cell. And the DNA is essentially organized on it into these loops. I hope you can see that. You can see that well enough. All right. Are there any questions about genomes and the way the genomes are organized and all that good stuff? All right. Well, if not, then we will soldier forth. And we will look at DNA replication. When Watson and Crick published their famous paper, they make a statement at the end of the paper, and this was very understated, but they said that the structure of this molecule suggests to us a means by which it might be replicated. The significance of this has not escaped our attention. So essentially, they suggested oh goodness, come on. There we go. They essentially suggested that if this molecule is double-stranded, you could replicate it essentially by unwinding these strands And then once you unwind these strands, you could essentially make new DNA in this fashion like this, using each strand here as a template for the synthesis of new DNA, so that each DNA molecule would have one old strand, so to speak, and one newly made strand. So this was the favored model. This type of model of replication is called semi-conservative. Semi-conservative because it keeps one old, makes one new. And while this was the predicted means by which DNA was replicated, experimentally, it was required that you demonstrate this. And there were some very elegant experiments that demonstrated this quite conclusively. And this essentially is the model that I showed you on that drawing, of course, drawn far better than I could. So you'll see these two are in this shade of purple. This one's over here. This one's over here. Then these two blue strands here are the two newly made strands, hence the semi-conservative nature of DNA replication. So you have complementary bases. A pairs with T, C pairs with G, adenine pairs with thymine, guanine pairs with cytosine. These are called Shoggoff's rules after Erwin Shoggoff, the chemist who showed the chemical relationship, quantitative relationship between adenine and thymine and cytosine and guanine by percentage weight in the molecule. So essentially, those two complementary strands will come apart. Each old strand will serve as a template for the synthesis of new strands. Those new strands are called daughter strands, and the parental DNA strands are called parental strands, like you see here. So you unwind the molecule like this, and then you make your new strands just like this. Now, the first organisms where DNA replication was studied in some detail were bacteria. Bacteria are easy to grow. They're very cheap to keep and grow in the laboratory, and they're relatively easy to do experiments with. Unfortunately, you can take bacteria and you can break them open, and you can give them things that they need, and the bacteria will, the extract from the bacterial cell will make DNA in the test tube. So it's relatively easy to study DNA replication using bacteria. Breaking them open is pretty easy to do. Making these extracts is pretty easy to do. And then you can add things, take away things from that extract to determine the enzymes that are required for DNA replication. So the first thing that became apparent with bacterial replication is, first of all, bacterial replication, DNA replication, is bidirectional. It occurs in two directions. Also, what became apparent is that there's only one spot on the bacterial chromosome where you initiate replication. So you initiate replication at one spot. It proceeds bidirectionally around the circle that is the genome until it meets at the other end, and then it stops and you have two bacterial chromosomes like this. So these types of structures are seen pretty commonly when you isolate replicating bacterial chromosomes. This is known as a theta structure because it kind of looks like the Greek letter theta. So this is a theta structure. You have two sites where replication occurs, here and here. there, right there. These sites here, where replication is occurring, those are called the replication fork. Replication fork. So on a bacterial chromosome, there will always be two replication forks. Replication will continue until you finish replicating these two chromosomes. These chromosomes might be interlocked, but then topoisomerase enzymes will resolve this and you get two separate bacterial chromosomes. The bidirectional nature of DNA replication in bacteria was demonstrated using this technique called autoradiography. This is a very time-consuming and very tedious procedure. Let me just explain it to you very briefly. So you get the bacteria so that they're dividing. So they're well fed, they're dividing, and then you want to try to get it so that all the bacteria are at the same stage in replication. The way you do that is you cold shock them. If you cold shock them, they'll stop the replication, and then when you bring them back to regular temperature, they'll start again. So all the bacteria are now replicating essentially that they've started replication. So you take your cells, grow them, cold shock them, heat them back up, give them everything, and you give them radioactive nucleotides for a little bit. And then after you give them those few minutes of radioactive nucleotides, you give them non-radioactive nucleotides, then you bust the cells open and you put the extracts onto a slide with a photographic emulsion. The radioactivity will expose the emulsion. You then have to put this into the freezer at -80 degrees for about a month. And the grains essentially will get exposed. You can see those grains here. And you can see those grains essentially over here. So you see these two places where replication is occurring, over here and over here. So this is a replication fork, that's a replication fork. This is telling you essentially you gave the organism these radioactive nucleotides. All of this gets labeled, all of that gets labeled, that gets labeled, that gets labeled a little bit, that doesn't get labeled very well. And that shows you essentially you have two replication forks essentially. That's autoradiography. So autoradiography definitively showed several times. And this shows you, this is from John Cairns' lab. It wasn't just Cairns, it was several other people who showed that DNA replication in bacterial chromosomes is bidirectional, always. The only time you don't get bidirectional replication is in some viruses, bacterial viruses, and in some plasmids. All chromosomes, at least that I can think of, are bidirectionally replicated. So the site where DNA replication begins is called ORI-C. ORI-C, which stands for Origin of Chromosomal Replication. As we said, there is one and only one ORI-C site on the E. coli chromosome. So if you look at the sequence of ORI-C from E. coli, you see these three things. And interestingly, these three things are found in ORI-Cs from most bacteria. There have been a lot of bacterial genomes that have been sequenced. They all have only one origin of replication. When you look at their sequences, you can find the origin of replication because they all have these particular features. So there's an AT-rich region. There's a sequence that binds this protein called DNA A. And then there's these GATC methylation sites, which isn't found in all bacteria, but it's found in E. coli. So here's OREC, essentially, from the bacterial chromosome. These are parts where this protein called DNA binds. And here are these AT-rich regions. You can see there's 3 of them. So there's 1, 2, 3, 4, 5 of these DNA binding sites. And there's 3 of these AT-rich regions here, at least in this portion of OREC. So DNA is a very interesting protein. It binds to these sequences here, but it also binds to each other. And it forms essentially this big ball of proteins. And the DNA of OREC is essentially wrapped all around this big complex of DNA proteins. Now, when that happens, the AT-rich regions, because the DNA is now under tension, they undergo strand separation. And then this protein called DNA-B will bind. DNA-B is a helicase. Helicases take DNA molecules and unwind them. So DNA-B forms a 6-subunit complex, like what you see here. So if we look up here, there's our DNA boxes, 1, 2, 3, 4, 5. Here's the AT-rich regions. DNA binds to them, it also binds to itself. This only shows 5 copies of the proteins. There's actually quite a bit more that binds. As it binds, it's going to take the DNA and put a substantial bend in it, as you can see. That bend puts tension on the DNA. To relieve the tension, the strands will start to unwind. DNA-B will bind here, and it'll start to unwind the DNA in this direction and in this direction. And these will become the replication forks. Now, those GATC methylation sites, what are those for? These are methylated by an enzyme called the DNA adenine methylase, which has the unfortunate abbreviation of DAM. Okay, guys, I don't name them, I just report them. So the DAM protein, that sounds awful, doesn't it? The methylase is going to essentially take this adenine and it's going to put on this adenine a methyl group. So it's going to stick a CH going to stick a methyl group, a CH3, on that adenine. What does this do? That methylation of the GATC site occurs after the start of DNA replication. Once it gets methylated, that tells the cell, okay, don't initiate another round of replication until essentially we get this methylated. And as you can see here, it takes several minutes for that adenine to become methylated. So you can very often have in cells several chromosomes that are being replicated. But so you don't overdo it essentially and get lots and lots and lots of molecules that are starting to be replicated that you can't finish on time. This regulates essentially the rounds of DNA replication. So only if the daughter strand is not methylated can it reinitiate. Once it becomes methylated, it cannot reinitiate. So initiation replication only occurs efficiently on that fully methylated DNA. So this replication essentially has to take time for this first round of replication to begin. So you have to make sure that the replication complex is far enough down chromosome before you initiate this again. That's essentially what the methylase does. So by methylating this, that tells you, okay, it's safe to start again. That's what the methylase does. So the helicase unwinds the DNA strands. It breaks the hydrogen bonds between the bases. The unwinding is going to increase supercoiling ahead of the replication fork. That makes it harder and harder and harder for the helicase to unwind the DNA eventually. So topoisomerase 2, also called DNA gyrase, goes ahead of the helicase, takes out those negative supercoils. And then after the strands are separated, these single-stranded DNA binding proteins bind to it to prevent those strands from reannealing. If the strands are reannealed, essentially the cell has just wasted a lot of energy unwinding it. To prevent that snapback, you have these proteins, so-called SSB proteins, that are going to bind to the single-stranded DNA and keep it single-stranded. Now, the enzyme that replicates DNA is called DNA polymerase. Very complicated enzyme. DNA polymerase is like a sprinter. So if you watch track and field, the distance runners don't use starting blocks. They can just start from the get-go. Sprinters, however, have to use starting blocks. DNA polymerase is a sprinter. It needs a starting block. The starting block comes in the form of little strands of RNA called an RNA primer. These RNA primers are anywhere from, here it says 10 to 12. They can be shorter than 10. They can be up to 8. 8 to 10, 8 to 12, 8 to 14, in that range, essentially. And these primers are made by an enzyme called primase. Those short little strands of RNA act as a starting block for DNA polymerase. So it gets to the free 3' end on that primer, and it will start then making a new DNA strand. So already you can see the number of proteins that will be involved with DNA replication, pretty substantial. So you have the DNA helicase, you have topoisomerase, you have single-stranded binding proteins, you have primase, and then you have the DNA polymerase. So the helicase unwinds the double helix that breaks hydrogen bonds between the bases that hold the DNA strands together. Topoisomerase 2, DNA gyrase, alleviates that supercoiling that forms because you're separating the strands. By alleviating that positive supercoiling, the helicase can do its job. Single-stranded binding proteins keep the strands from reannealing, keeps them apart. And then the primase will synthesize an RNA primer. So DNA polymerase can get a start. The primer acting kind of as starting blocks, if you will, for DNA polymerase. Now, in bacteria, the main DNA polymerase is called DNA polymerase 3. DNA polymerase 3. DNA polymerase 3 is the workhorse for DNA replication. There's another DNA polymerase that works as a repair enzyme and does some work in DNA replication because it takes out those RNA primers and replaces them with DNA. That DNA polymerase is called DNA polymerase 1. DNA polymerase 1. When the RNA primers are replaced with DNA, there's still a gap in the DNA backbone that the polymerase cannot fill. That nick in the backbone is filled in by DNA ligase. It will link them together. Now there's a term here, and this will be the last thing we'll talk about. We'll close here. Okazaki fragment. Okazaki fragment. These RNA primers, when they start having DNA added to them, that fragment, the RNA primer with the DNA attached, is called an Okazaki fragment. It's named after a husband and wife team who are at the Tokyo University of Technology. Meiji and Ayako Okazaki, and they discovered them. That's why they were called Okazaki fragments. So that's what that term means. And this statement here is actually not completely correct, because when the ligase links those fragments together, they don't have any RNA in them, so they're no longer Okazaki fragments. So this statement is actually not completely correct. It links together, it fills essentially the gaps in the fragments. So this illustrates that here. We'll talk about this. We'll look at a video of DNA replication from YouTube that kind of tells the whole story. And that is essentially all for today. I'll hang out here for just a bit. After we've finished, if you wish to ask any questions. And I can't hang out for too terribly long because I have another class in 10 minutes. So I will bid you all guys all a fond afternoon. I will see some of you, well, almost all of you tonight. So have a good afternoon. And again, I'll hang if you want to ask any questions.
I have a question. So
Yeah.
I know that it says in the syllabus we are just going over like the slides, but I was just wondering like if there's anywhere in the book that I could go and read extra over what we're talking about.
No, but there's a site that's run by the journal Nature that has a lot of really good basic stuff about DNA replication. I might have put it in the link. I don't know who is doing this, but some idiot keeps trying to call me, and I keep blocking these people, and they keep recalling me. It's really annoying. Okay, let me check the website here, the Blackboard site here, just a sec. No, I don't have that site up yet, Natalie. I will get that site and I will put it online. It's an educational link from the journal Nature, the really famous British journal. I'll find it. I'll put it in Blackboard. That has pictures and all kinds of nice stuff written by experts in the field. I'll get that on Blackboard. Okay.
Perfect. Thank you so much.
For the farm quiz later, do you want us to do that in class or can we do that anytime before it's due?
No, we're going to do it in class. You might have noticed there's a password.
Oh,
required.
okay.
You can't get in with that password. I'll give you the password in class. Well, at our Zoom class tonight. You guys will then just do it, hand it in, and it's just a quick Blackboard test. Shouldn't take you long at all.
Okay.
Okay?
Thank you.
You're very welcome.